Hi, everyone. I'm Ananya. I'm from the Bay Area, California, and I am presenting on the impact of BRCA1 and BRCA2 mutations on metastatic breast cancer. So I'll be going over the genetic causes of breast cancer, the structure and function of BRCA1 and BRCA2, BRCA1 and BRCA2 mutations, and how they are connected with breast cancer, findings from my research, and discuss those findings. So for a little bit of overview, we're going to go over some genetic causes of breast cancer. The most significant risk factor for breast cancer is a positive family history. And the genes that are linked with high risk for breast cancer are BRCA1, which is on chromosome 17q21, BRCA2 on chromosome 13q12.3. And another gene that is not really covered in my research, but it is something that to consider, is TP53, which is also linked with earlier breast cancer development, specifically with Li-Fraumeni syndrome, which is a disorder that increases one's lifetime probability of developing cancer. So we'll be specifically focusing on BRCA1 and BRCA2, but TP53 is important background. In order to understand why BRCA1 and BRCA2 can cause breast cancer, it's important to understand what DNA repair looks like. So DNA is a double-stranded helix, and specifically when BRCA1 and BRCA2 come into play is when there is a double strand break, when both strands of the DNA are broken at one spot. As you can see in the top here, BRCA1 already plays a role in which mode of repair the cell decides to do. And then we'll find later on that BRCA2 is really impacted with how RAD51, a different protein, binds onto the single-stranded DNA once it's broken, so that proteins can come in and recreate the section that is missing. And this is called homologous recombination repair. And the presence of BRCA1 really dictates whether the cell decides to go through non-homologous end-joint repair, where the two sides are just stuck together, or homologous recombination repair, where it is resynthesized, the parts that are missing. So this is a more in-depth picture of what homologous recombination repair looks like. So we get a double strand break, and then BRCA1 comes in and causes homologous recombination repair to happen in the first place. The break is repaired. The reason why it's called homologous recombination repair is because a homologous DNA strand, which means a similar DNA strand, is used as a template. So as you can see here, there's a sister DNA strand that is captured, and DNA is synthesized according to this blue template. And that is how the cell recreates the break and makes it so it's fixed. BRCA1 and BRCA2 play critical roles in this process. BRCA1 here starting the whole process, and BRCA2 bringing RAD51, which is a really important protein that helps create these, helps start off this DNA synthesis process. So let's dive into BRCA1 a little bit. This picture down here is what BRCA1 looks like, and these are the two sections we'll be focusing on. So BRCA1, the gene, encodes a protein, which is also called BRCA1, with 1863 amino acids. The specific notable regions are an acidic C-terminal region called the BRCA1 C-terminal region. This is important. It's called the BRCT region, and that's right here. And there's also the N-terminal ring domain, which stands for really interesting new gene. So BRCA1 is a protein that binds BARD1, and this protein is a little similar to BRCA1. Known mutations of BRCA1 that can no longer bind BARD1 are linked with cancer. So what this tells us is BARD1 also has an important role to play in this DNA repair process. And without BARD1 working properly, that can cause cancer. And this also tells us that that BARD1 is significant for suppressing these tumors so cancer doesn't show up in the first place. Additionally, there's a part in the BRCT region that can be used as a p53 binding site. And p53 is known for regulating the cell cycle and apoptosis, which is cell death. So what this tells us, and apoptosis is important because the thing with cancer is the cells don't die when they are damaged. So they just continue to grow and grow and grow. So if P53 isn't able to do its job, that can influence whether a cell undergoes apoptosis or not. So we can see that BRCA1 plays a really important role in a lot of different functions, including like how DNA is repaired, but also potentially within regulating the cell cycle, and whether a cell dies or not when it is damaged. BRCA1 is involved in regulating both homologous recombination and non-homologous end joining repair, as we saw in the first slide, where its presence dictates which of the two pathways the cells undergoes. And then BRCA2. There are a few parts we're going to be going over in BRCA2. These are the BRC repeats, the OB folds, and the helical domains. So the notable regions are the BRC repeats, the DNA binding domain, which is right here, and the C-terminal TR2 domain, which is right here. So in case of the double-strand break repair via homologous recombination, RAD51 is essential, as I mentioned earlier. And these BRC repeats is what binds RAD51. And without RAD51, it's not able to form the filaments needed to hold the single-stranded DNA in place when the synthesis, when the cell's trying to synthesize DNA. So BRCA2 stabilizes these RAD51 filaments with this domain right here. So what's important is that BRCA2 is a protein that binds with RAD51 at multiple points. So when it binds with RAD51 here, and then it stabilizes here. So without these two portions, RAD51 isn't able to do its job. So the C-terminal region here is really important for the homologous recombination repair, and cells without the C-terminal region are more sensitive to radiation and DNA damage. When DNA is broken, it's really important that cells are able to repair it without damaging the cell more. We want the DNA to be functioning and we want it to be correct. So without this area, the DNA is more damaged, which can be a really significant problem. And then here with the single-stranded DNA, so this part binds to the RAD51, and this part binds to the single-stranded DNA, and therefore causes RAD51 to be able to form its filaments on that template strand, which is exactly what I said. So yeah. And then let's go over how the mutations can cause breast cancer. We went over this a little bit with which mutations are linked with them. So for BRCA1, the domains that are mutated in high frequency in breast cancer patients are the ring domain, this region between exons 11 to 13, which I'll talk about later, and the BRCT domain. So the ring domain mutations, because they have an increased risk of breast cancer, it suggests that the ring domain has an important role in suppressing the tumors. So what's important to know about these, this research is you really can't say this mutation, like we know that this part isn't important for this reason, and therefore if it's gone, it'll cause mutation. We have to look at which mutations are common in cancer patients and then work backwards from there, which is why a lot of this is, it's working backwards rather than going from this thing is important. Instead, we look at this mutation is common, so it must be important. For exons 11 to 13, right now its function is not really known, but mutations in that region is also very highly linked with breast cancer. And again, the BRCT domain, because it is very commonly mutated in cancer-associated mutations of BRCA1, it must have an important role in suppressing tumors or making sure that cancer doesn't develop. And then going over BRCA2, mutations affecting that BRC repeat structure, which weakens the RAD51 binding, is associated with cancer predisposition. Deleting the C-terminal region is associated with early onset breast and ovarian cancer. And cancer-associated mutations in the DNA binding region can also cause defects in homologous recombination repair. So what this tells us is just that BRCA2 has a really important role to play with RAD51 specifically. Another gene that another protein it's involved in is this PALB2. And what this does is it recruits BRCA2 to double-stranded breaks. So we're seeing that PALB2 recruits BRCA2. So we're seeing that this other protein can also be involved in this DNA repair process. And mutations in either BRCA2 or PALB2 that prevent this interaction is also linked with cancer. So there's a lot of mutations in both of these genes that can cause the proteins to not do their job. Another important part to go over is the subtypes of breast cancer, and then we'll get into findings. So breast cancer has a few different subtypes based on these different hormonal receptors and whether they're present or not. And these are the 4 subtypes that are present: luminal A, luminal B, HER2 positive, and triple negative breast cancer. So when we have these cancers, we can see that triple negative is very aggressive, and it's very difficult to treat, while the luminal A and luminal B cancers are a bit easier to treat, as in they have a better prognosis. And HER2 positive breast cancer has specific therapies for it because it has different receptors that need to be treated. And then how these are connected with metastasis, we can see that luminal A, luminal B, and TNBC are linked with higher rates of brain, liver, and lung metastasis, and TNBC has a lower rate of bone metastasis as compared to the luminal ones. And we can see the best and worst survival here on the right. So triple negative is very aggressive and very dangerous. So our findings, we found through my research that BRCA1 mutation carriers usually result with a TNBC subtype, which is, as we went over, associated with aggressiveness and a high tumor grade, which basically means more difficult to treat. And it has a predisposition for central nervous system metastasis. While BRCA2 mutation carriers usually have luminal-like subtype, especially luminal A, which is associated with a more positive prognosis and slower growth. And what we can tell from this is when we are saying that BRCA mutations have a significantly higher frequency of CNS metastasis, stage 4 cancer, we need to think about what does this mean for treatment and what does this mean for what patients are able to do. What this tells us when it's saying that BRCA2 mutation is the only one that's significantly associated with CNS metastasis when controlling for cancer subtype is that BRCA1 leads to TNBC, and TNBC is connected with CNS metastasis. Whereas for BRCA2, regardless of whatever subtype it is associated with, whether it's luminal, like it most commonly is, or any of the others, it is in and of itself associated with CNS metastasis. So this is really important for knowing where cancer is likely to spread in a patient. BRCA1 carriers more frequently experience lung and distant lymph node metastasis. So what this information is telling us is it's telling us patients with these mutations are more likely to go through metastasis in these certain parts of the body, which is really important for treatment. And then BRCA2 carriers and non-carriers experience more bone metastasis than BRCA1, which is also linked to TNBC having less bone metastasis than the other subtypes. So we have, overall, to summarize, BRCA mutations are linked with CNS metastasis in breast cancer patients. We know that impaired DNA repair can contribute to genomic instability or damaged DNA, which can cause certain types of metastasis to occur. And then what we need is larger, more comprehensive studies to confirm these findings and find out how BRCA mutation subtypes and metastasis are interconnected. Like, for example, BRCA1 mutations linked with TNBC means that there's a more aggressive and a higher metastasis risk. So future research should clarify these pathways and focus on how other components in the homologous recombination pathway can also involve themselves in cancer progression, so we can develop more individualized approaches to improve outcomes for patients afflicted with BRCA-associated breast cancer. So what we have overall is understanding the connection between BRCA mutations, breast cancer subtype, and metastatic patterns is crucial for improving risk assessment, early detection, and overall patient outcome. Thank you. And some of my references.
Wonderful. Thank you, Ananya. That was great. Is like the BRCA mutations, is that a common thing that they test for?
It depends on how the cancer is showing itself. It's not really very common also because genetic testing in and of itself is expensive, and it takes time. So there's also a lot of equity issues around this. So there's a lot to do more research on and improve ways to be able to test for this.
Yeah, well, I mean, the research was super informative. Was there like an aha moment for you when you were going through like it was a big learning for you as you were going through the project?
I think one of the main things was I did not expect like so many different parts of cancer to be associated with these mutations because I didn't know about breast cancer subtypes and I didn't think they would be like significant enough to mention. But then it turns out that BRCA mutations are very much significantly linked to subtypes, and then subtypes are connected with metastasis.
Yeah.
So yeah.
Yeah.
Also, there's like not much like what I had when I was going through my research, I had to like cherry pick a lot of different sources because not a lot of them specifically targeted these connections. So I had to like find a bunch of different sources and then connect them all.
Had to piece it together yourself, right?
Yeah.
Yeah. Well, it was very well done and very informative. I appreciate it so much. Thank you, Ananya.